Smith from William and Mary, and this is the second part of Lecture 9 of Cellular Biophysics and Modeling. So Lecture 9 is a combination of two topics. We're discussing voltage clamp recording, and which is a new recording mode for us, as distinct from current clamp, and we're discussing the classical biophysics of the squid giant axon. The figures from this lecture are drawn from the textbook Cellular Biophysics and Modeling, a primer on the computational biology of excitable cells. It's chapter 12 and chapter 13 that you should be reading in addition to listening to this lecture. And a number of the figures I'll be showing are from this classic book by Bertil Hille, Ionic Channels of Excitable Membranes, which comes highly recommended. All right, to review part one of this lecture, there are two very different recording modes that we are actually discussing in parallel right now. So what we've done for the first unit of the class is primarily focused on current clamp recording, and there's a current balance equation for the cell membrane potential that we've been discussing and deriving and solving. And the type of experiment that this equation can help us simulate or model, understand, are these experiments where the experimentalist has control over the applied current as a function of time, and then the recording is of the changes in the cell transmembrane potential that are occurring as a function of time. And one of the things that was important to note is that when the applied current is positive, that's actually a depolarizing influence on the cell membrane potential. But it kind of goes without saying that for the entire unit prior to this one, the applied current has been sort of the independent variable, and the voltage has been the dependent variable. So the applied current has been dialed in by the experimentalist, and the experimentation is recording, right? The electrical equipment is recording the voltage as a function of time. In voltage clamp recording, the picture is reversed. So in this case, it's a target or command electrical potential that's dialed in by the experimentalist, and the electronic apparatus is injecting whatever amount of applied current is necessary in order to maintain that command potential as a function of time. And in the last lecture, we explained that most of the time, with the exception at these moments when the command potential is jumping up or being returned to rest very quickly, most of the time the rate of change of the membrane potential is zero here, right? And if you think about what that means from the perspective of the current balance equation, the applied current that's necessary to maintain the command potential when the rate of change of the voltage is zero is actually a measurement of the ionic currents that are flowing through the cell membrane. Now, the membrane currents include capacitive currents as well, and so that's what is responsible for these vertical spikes or downward deflecting spikes here. And there'll be more discussion about that phenomenon in the textbook, and I'm going to just put that aside for now so that we can move on. But essentially, these capacitive transients reflect the need to actually apply current to charge up the membrane to depolarize it or to repolarize it. So it sort of makes sense. The voltage is being depolarized here, so there is a applied current that has to be very strong and very positive initially, and the reverse on this edge. So you've seen this sort of picture before, I'm sure, from introductory neuroscience courses. We think of the cell action potential as having a number of phases. There's the resting membrane potential. If you depolarize the cell a little bit, you might go over this threshold, at which point the opening of voltage-gated sodium channels sort of has a runaway dynamic where inward current creates more inward current, and so the membrane potential jumps up, sometimes even above 0 millivolts. And then there's a repolarization phase where the sodium channels close and potassium channels open, and the membrane potential comes back down to rest, and it often will undershoot. And that undershoot phenomenon is sometimes called the after hyper polarization. Now, neurons exhibit all sorts of electrical responses, and at this point, I just want to survey a few of the sorts of responses that are of interest to us as neuroscientists, because just to motivate what we're going to be doing in the rest of this lecture. So there's sort of two related phenomena. One is the action potential itself, which is a form of what dynamicists call excitability. And repetitive spiking of cell membranes is an example of neural oscillations. So we'll be going back and forth between these two phenomena ultimately. You can see that the action potential here in Purkinje neurons is slightly different than in pyramidal neurons, which is different than in dopaminergic neurons. So there's all sorts of subtle differences between the action potentials that are exhibited by these neurons, and the repetitive oscillations that the membrane may go through also have different dynamics. This is a sequence of current injection. So this is current clamp mode where a little bit of current, 2 picoamps, gets the neuron to start spiking, and you can see that the spiking frequency increases as the amount of current being injected increases, but eventually you sort of overwhelm the neuron and you lose the spiking phenomenon when you inject too much current. This is summarized in something called a current frequency relationship. So what we mean is the injected current, the applied current, is on the horizontal axis, and on the vertical axis is the firing frequency of the neuron. And in this experiment, shown on the left, you can sort of see this, looks like sort of a sideways parabola, giving the increasing firing frequency as a function of the applied current. And but notice that, you had to inject a little bit of applied current to get this thing to spike at all, so it might not have been spiking at 1 picoamps. And it's possible that the steady state response will be non-oscillatory if you have too much current. So this right here at 40 picoamps, this is the initial firing frequency, but that wasn't maintained permanently in these experiments. It only did this a couple of oscillations, and then the action potential ceased. Another thing to note is that muscle cells are also electrically excitable. In particular, cardiac myocytes exhibit action potentials that are responsible for the electrical signaling of the heart, and but these action potentials are much longer in duration, and we will postpone our discussion of them for a little while. But the neuronal action potentials tend to be just a few milliseconds in duration, whereas cardiac action potentials can be hundreds of milliseconds in duration. Okay, so we're going to study this phenomenon of action potentials, or membrane excitability, and repetitive spiking, or neural oscillations, in several ways. Firstly, we're going to show how it was historically observed in classical experimental preparations. And then we're going to also discuss the membrane models that were produced to help understand those experimental results. The two cases that we're going to consider are Hodgkin and Huxley's classical experiments with the squid giant axon. And in this case, the currents of interest will be a passive leak, a voltage-dependent sodium current, and the voltage-dependent potassium current that's sometimes called the delayed rectifier potassium current. We'll also discuss excitability and oscillations in the context of barnacle muscle fibers, and the model in that case that was developed to explain those oscillations is called the Morris-Lacar model. The interacting currents in that case are the leakage current, a voltage-dependent calcium current, and the delayed rectifier potassium current. So similar combinations of currents leading to similar phenomenon, and both of these are important historically. So, what were Hodgkin and Huxley's contributions? Basically, these Hodgkin and Huxley got the Nobel Prize for their experimental observations of action potentials in the squid giant axon and the mathematical modeling that they developed that included a current balance equation, representations of voltage gated ionic currents, and so on. And we'll be talking about that modeling as well as the experiments here. Of course, it goes without saying that we're not talking about giant squids. We're talking about normal-sized squids and giant axons that are in the stellate nerve of these squids. These can be dissected out, and the reason why they're so large is that the size, the diameter of the axon is related to its conduction velocity. We'll get to that down the road. And this is part of an escape reflex. This nerve is part of an escape reflex where it's been adaptive for the signals to be extraordinarily fast, and that's what's led to this phenotype of having a very, very large axon, meaning large diameter axon running down the length of the squid. So if you dissect out a couple millimeters to centimeters of this and axon and then run a wire through it, you have a nice preparation of an excitable membrane that you can stimulate and record from. Now, when this was done, there was a primary observation, which was that the action potentials that were evoked by electrical stimulation could be suppressed in amplitude by decreasing the sodium concentration outside the cell. So 1 is control, 2 is with reduced sodium in the extracellular media, and then 3 is with the sodium concentration returned to normal. And this strongly suggests that it's actually the influx of sodium somehow that is important in the generating the action potential. Now, something that I'm going to need you to do in the future is when you see a figure like this, you're going to need to decide, is it current clamp or voltage clamp recording that we're looking at? This isn't always obvious at first glance, but it's very important to sort that out in your head before you start analyzing the figure. So we have no indication of what the applied current is as a function of time. So it's probably 0 or constant. And this S here is standing for stimulation. So this was some sort of electrical stimulation leading to the action potential. And so we would view this as a current clamp recording in the sense that the input was a current that was under experimentalist control, and the recording itself is a voltage that was the response, or the sort of the dependent variable in the experiment. This is an example of a voltage clamp recording. So you look at this, and the first thing that you have to figure out is that it's not current clamp, it's voltage clamp. And I can sort of see that because if you go to panel B, for example, this is a membrane potential that's being sketched, right? And it's starting at minus 65 millivolts, and then at time 0, it's being stepped up to 0 millivolts. And this fact that it's a sort of square pulse like this suggests that this was under the experimentalist control, and what was being measured was the membrane currents flowing as a function of time. Initially 0, and then there's a phase of inward membrane current, followed by outward membrane current. So this was one of the observations of Hodgkin and Huxley, this biphasic response. And remember the downward deflection here represents the measurement of a membrane current, and negative membrane currents are inward, whereas outward membrane currents are positive. Let me say that again. Membrane currents that are negative in sign, right, are actually inward membrane currents. Membrane currents that are positive in sign are outward membrane currents. So this would be a depolarizing influence here, and this would be a hyperpolarizing or repolarizing influence here. Okay, so these two phases of membrane current are exhibited after this, in this voltage clamp recording, after a step in the command potential from minus 65 to 0 millivolts. If you start at minus 65 at rest and you drop the electrical potential down to minus 130 millivolts, there's very little current that flows. So clearly there's some sort of voltage dependent opening of ionic channels in this excitable membrane, and the direction that the voltage is being stepped makes a big difference with regards to whether those currents flow or not. Now, because you can reduce the size of the flux of inward sodium current by manipulating the extracellular sodium concentration, Hodgkin and Huxley were able to reproduce this voltage clamp experiment in the context of smaller concentrations of extracellular sodium. And when they did that, they got a current. The current that was evoked by depolarization was uniphasic. It was a sigmoidal function here that was positive or outward. So the inward phase was eliminated by reducing the extracellular sodium concentration. So the interpretation was that this biphasic response is a combination of sodium and potassium currents, in that when you eliminate the sodium current, you get the outward potassium current. And so if you subtract these two curves, this is the dynamics of the voltage-gated sodium current. Now, in order to summarize experiments of this type, you could plot what they refer to as the plateau current for potassium and the peak value of the sodium current. So what do we mean by that? So here, for the sodium current, it's inward for a certain amount of time, and then it goes back to zero. And so if you're going to if you want to plot the value of the sodium current as a function of the step voltage, you need to use the peak amount of inward current. So the nadir of this biphasic curve here is the sort of maximum amount of inward current that flows in response to this depolarizing step in the command potential. And in the same way, because the outward potassium currents sort of turn on and remain on, we can plot the value of this current that persists in that case. So if you do this over and over again, the plateau value for the potassium current looks like this outward rectifying curve, and the current voltage relationship that's as measured using voltage clamp recording for the sodium current has this form that we've been discussing previously in class. Remember that this type of current here is restorative, and this one is regenerative, and it's the fact that the sodium current is regenerative that's leading to the upstroke of the action potential. Now, one of the things that Hodgkin and Huxley realized is that they could back calculate the sodium and potassium conductance as a function of time under the assumption that the current was the product of a conductance and the driving force that we've been writing, the voltage minus a Nernst equilibrium potential. So if the current is assumed to be a conductance times a Nernst equilibrium potential, then if you rearrange those terms, you can get the conductance is the measured ionic current divided by the driving force. And when that is done, you can see the sodium current turning on here and turning off again, and the potassium current turning on but persisting. So now you might say, but wait, sodium is inward. Why is the deflection upwards here? And that's because we're plotting the conductance. The direction of the current was sort of factored out by dividing by V minus ENA. So conductances are positive or non-negative. And so when you plot the sodium current conductance, it's a biphasic positive curve, whereas the potassium conductance is sort of the sigmoidal positive curve. Now, if you do this over and over again, right, for different values of the step potential, you get all sorts of different dynamics of membrane current. It looks very nonlinear and very complicated. And one of the sort of genius aspects of what Hodgkin and Huxley did was they assumed, let me see here, they assumed they assumed that the measured membrane currents were going to could be sort of well represented by a current balance equation of the type we've been considering, with this addition of these scalar quantities that are referred to as gating variables. So what I mean is, here's if the normal current balance equation that we've been looking at for a passive membrane, it includes the capacitance times the rate of change of the voltage, the applied current, and then like a leakage current, which might be mediated by potassium and a little bit of sodium. But this is sort of the starting point for our understanding of the resting membrane potential. And what Hodgkin and Huxley did was they're adding representations of the voltage-gated sodium current and the voltage-gated potassium current into the current balance equation. Now, these are modeled in this fashion that we've been doing, where there's a conductance times a driving force, but they're introducing another variable here, which can take values between 0 and 1. And in this case, I guess the language that was used, as they called them, gating variables. So they're dimensionless numbers that can go between 0 and 1, and you can think of 0 as being these gates are all closed, and 1 being the gates are all open. There were 2 gates that were used to describe the sodium current, and we'll go into that in some detail in a second. The point is that those experiments could be used to fit curves for the steady state value of these gating variables as a function of voltage, and also for the time constant of changes for these gating variables as a function of voltage. And in that way, they were able to produce a experimentally sort of tuned phenomenological model of the electrical potential changing as a function of time. So let me go back to I'll go here. Right, so they have all these measurements of how the conductance is changing as a function of time, and it's from those that these properties of the hypothesized gates for the ionic currents are back calculated. Now notice the sodium activation gate is an increasing function of voltage, whereas the inactivation gate is a decreasing function. The potassium current's activation gate is an increasing function of voltage. And the time constants tend to be larger at intermediate voltages and smaller at extreme voltages. So if you take the model that Hodgkin and Huxley produced, and you just look at the dynamics of these gating variables as a function of time, you get sort of an understanding of how the ionic channels of these excitable membranes are opening and closing in response to the step in the command voltage. So for example, in the case of the potassium current, the step in the command voltage is causing this gating variable N that was initially 0 when the cell was at rest to relax to a much larger value. Let's go back and see if that makes sense. So here's the gating variable for N. When you're at rest, N is at its steady state is near 0, but when you step to higher voltages, N wants to relax to a new steady state, which is closer to 1. The sigmoidal aspect of this, the current that's evoked due to depolarization, is represented by taking this gating variable to the power of 4. So the Hodgkin and Huxley presumed that the potassium current is some constant conductance for these channels times the fraction of these gates that were open to the fourth power times a driving force, and that fit their experiments quite well. It's a little bit more complicated, but they determined that in order to model this transient sodium current, they needed more than one gating variable. M is the activation gating variable, H is the inactivation gating variable. So at rest, M is near 0 and H is near 1. Okay, at rest, M steady state is near 0, whereas H steady state, right, is near 1. And then when the depolarizing current pulse happens, the first thing that you see is that M rapidly increases to its new steady state, and the H gating variable decreases exponentially to its new value. It's the combination of M to the third power times H that gives you this biphasic response in their model. Notice that for both M and N, right, values of 0 mean no current flowing, and values of 1 mean current will flow at the maximum rate. And that sort of makes sense. So then that variable, you can think of it as representing sort of the fraction of the gates that are open. On the other hand, H is a inactivation gating variable, and the inactivation itself is represented by a change in this gating variable, and then going towards 0. It's starting at 1 and it's decreasing towards 0. So the current's only going to flow if M is near 1 and H is also near 1. So this is leads to some terminology that we need to get straight, which is that when we have an activation gate like M or N, these gates are relatively fast compared to the inactivation gate. And when these gates go from 0 to 1, that's gonna be representing the process of activation, or the opening of these ion channels. And then when these gates return to 0 from 1, we refer to that as deactivation. On the other hand, for the inactivation gates, the process of inactivation, which is closing of the ion channels into a long-lived closed state, is a decrease in the gating variable, and then the recovery is an increase. And these two phenomenon are referred to as inactivation and de-inactivation. So the language, this is very standard language, and it's a little bit confusing at first. Just think of activation is just opening of the channels. Deactivation is the opposite of that. So it's closing of the channels. But there's also another possibility that the channels can be inactivated, which means they're transitioning into a long-lived closed state. When that process happens, it's called inactivation. And when that process is relieved or wears off, it's called de-inactivation. So the DE prefix always means the opposite, or the recovery of the variable. So here it is again. This all that work leads to this Hodgkin-Huxley model of the action potential in the squid giant axon, and this was really a tour de force when these experiments were performed and this modeling framework was produced. It's important to remember that I guess the experimental preparation had been used before by others, but they modified it, improved it, and they did all of the experimental work that was necessary. But in addition, as though that weren't enough, they actually developed this whole modeling framework, fit the parameters of the model so that they can reproduce what they saw in their experiments. And they were doing this at a time when there were no microcomputers. Essentially, you were integrating these ordinary differential equations by hand using a numerical technique called Euler's method. And it was a laborious process of taking each time step by hand and calculating how the gating variables and the voltage would change as a function of time. Now, this Hodgkin and Huxley model of the action potential is quite famous at this point, and you can find online all sorts of different um, simulators, and these simulators essentially are integrating the Hodgkin-Huxley ordinary differential equation system. So here, there's a simulation of a current clamp recording where the cell's at rest, a depolarizing pulse evokes an action potential. And here, a hyperpolarizing pulse actually causes the membrane potential to hyperpolarize a little. And when that pulse is returned to zero, you get another action potential. One of the things that this um, um, the modeling helped Hodgkin and Huxley understand were counterintuitive phenomenon like this, where an excitable membrane would have sort of a backwards response to the injected current. And this phenomenon goes by the name of the post-anodal break excitation. So anodal referring to this negative current pulse. And you can see in the equations here for M, H, and N that the model is suggesting that this occurs because this hyperpolarization here is actually causing the inactivation gating variable for the sodium current to move towards 1. So the sodium current is becoming less inactivated during this hyperpolarizing phase, and there's something about that causes there to be spontaneous action potential to occur after the cell membrane, after the applied current is returned to rest. Here's another example of a simulator that I found online. And what's being shown is the injected current pulse here in the positive direction. That current pulse is causing the membrane to oscillate. And you would be able to, using a simulator like this, construct a current voltage relationship, a frequency current relationship where this would be the applied current on the horizontal axis, and then you would take the period here of the oscillation evoked, take its inverse, and that would be the function of the firing frequency as a function of the injected current, which we saw in the experiments at the beginning of this talk. I suppose it has to be the case if they fit all these gating variables, right, to the results of their voltage clamp recording, that when you put everything together and simulate the Hodgkin and Huxley model, you actually are able to reproduce the voltage clamp responses. And this is an example of from the textbook where the Hodgkin-Huxley model is simulated in the voltage clamp configuration, and you can get the biphasic response to a depolarizing command potential that was one of the initial observations that Hodgkin-Huxley made. So now we understand, or we have a representation of how complex membrane current phenomenon might be evoked by changes in the membrane potential. The idea here, there's more about this in the textbook, but the point is now that anytime you can use the Hodgkin-Huxley equations to reproduce an experimental phenomenon like triggering an action potential, and if you are interested in like what determines this, the depth of the after hyperpolarization, or what determines the latency between the time of the stimulus and the upstroke of the action potential, you would have the model actually gives you ideas about why that might occur, because the model is showing you these hypothesized gates and how they're changing in response to membrane voltage. And the model can also tell you how the currents that are flowing inward and outward are changing as a function of time. So it allows you to get sort of see the inside dynamics, see things that an experimentalist wouldn't be able to directly observe. So this would be a pretty sophisticated hypothesis of what's going on during an action potential, and such a, this, if you think of this as sort of a quantitative hypothesis for how injected currents are causing ionic currents to flow, any response that you see here, if it reproduces the experiment, you have a suggestion as to why that it's working the way why the experimental result actually occurred. And if there were experimental phenomenon that were exhibited by the actual membrane that the model wasn't able to reproduce, it would suggest that your understanding that you that sort of determined how the model was set up was off, and that you might want to augment or adjust the model in some way. So several ideas of the sorts of phenomenon associated with action potentials that can be understood using the Hodgkin-Huxley equations are shown here. Depending on the size of a current pulse, there's a different latency of the response to the first action potential. So that phenomenon can be understood by considering the dynamics of the gating variables in the Hodgkin-Huxley model. And there's also a phenomenon of refractory period that can be reproduced by the model. So in this case, there's a stimulation, an injected applied current leads to an action potential, and a second pulse leads to a second action potential, as you might expect. But if the second pulse is coming too soon, this following action potential doesn't occur. And that would be, that's what people are referring to as a refractory period. The membrane is excitable, but it's refractory or not excitable for a particular amount of time after the action potential has occurred. Here's the understanding of how post-anodal break excitation occurs. So even here, the hyperpolarizing current pulses, right, are causing the membrane potential to go down, and it's during that phase that the gating variable for the inactivation of the sodium current goes up, which means that you're actually generating more sodium current through this hyperpolarization event, and that increase is what's allowing the membrane to spike after the applied current is returned to zero. Here's superthreshold applied current causing repetitive action potentials. You're showing the dynamics of the gating variables, showing the dynamics of membrane potential, and you can look in the model and ask about the dynamics of the membrane currents that are both inward sodium and outward potassium. Now, when you want to summarize these sorts of simulations, what you might do is very much like what we started with, construct a frequency current relationship for the membrane. So here's here I've done that using the Hodgkin-Huxley model, where I'm applying current steps to various values and exhibiting oscillations at various frequencies until I'm injecting so much current that the oscillations cease. So this phenomenon here is referred to as repetitive spiking or oscillations. And if you inject so much current that you don't get any oscillations at steady state, that's called depolarization block. You can summarize a set of experiments or modeling simulations of this type in a diagram that's a type of bifurcation diagram, I suppose, where the applied current is the bifurcation parameter, right? That's the one parameter that's being changed here. And on the vertical axis, we're plotting either the steady state membrane potential that's exhibited, right? So at very low currents, the steady state here is like minus 80. At very low currents, this is actually negative applied current, and that's why it's below rest. But this dot here corresponds to that steady state voltage, which corresponds to that steady state voltage, and then so on. As you increase the applied current, you see that steady state can change, and you also are plotting the maximum and the minimum value of the membrane voltage during the oscillation. So now, if you think of this bifurcation diagram here, where you're changing the applied current, and instead of the minimum and maximum voltage, you plot the actual firing frequency, you get this curve that looks like a sideways parabola. And one of the details that ends up being pretty important here is that for this model membrane, the spiking starts at a particular frequency. So you can't sort of get the neuron to spike and then back the applied current off and have the neuron spike arbitrarily, slowly. At some frequency, it just stops spiking, and what that precisely what that frequency is an important property of the cell membrane that we're going to talk about in a future lecture. Okay, so where do we go from here? So using this Hodgkin and Huxley style modeling, where the membrane conductances that are hypothesized are assumed to be proportional to the product of some number of gating variables whose dynamics are controlled by first order kinetic equations for their steady state and their time constant as a function of voltage. That's sort of the framework that Hodgkin and Huxley developed. We can simulate and compare and contrast models with experiment to our heart's content now. So we'll be doing a lot of that. But a mathematically minded person might be concerned that the simulations that we're doing are still pretty complicated, nonlinear ODEs. There's 4 variables to consider. And so it might be hard to understand why the dynamics of these gating variables are leading to the membrane potential phenomenon that's being exhibited. And one of the reasons why we're going to look at this Morris-Carr model for the excitability in barnacle muscle fibers is that this excitability in this system is a little simpler to understand. It can be modeled with 3 as opposed to 4 variables, and there's even a trick that gets you down to 2 variables. And if we understand the Morris-Carr model of excitability in barnacle muscle fibers, then we have a nonlinear differential equation system that has only 2 variables, and there are techniques, graphical techniques that we will investigate called phase plane analysis that will give us a lot of information about what's really going on when we sort of drill down into the nonlinear dynamics of excitability and oscillations in these membranes. So things down the road that we'll be talking about are to help us deeply understand membrane excitability and oscillations include phase planes, a technique called linear stability analysis. And then just like we did bifurcation theory for one-dimensional systems, we'll now start talking about bifurcation theory for 2D systems. And then ultimately, I'll give you some work to do with the so-called Fitzhugh-Nagumo model, which is a 2D nonlinear dynamical system that sort of has polynomials on the right-hand side, but it exhibits many of the phenomenon that both the Hodgkin and Huxley model and the Morris-Carr model can produce. And that will be a model that we can really open up the hood and see exactly what's going on, because the right-hand side of the differential equations is so simple. So just one final note, there is a justification for the Hodgkin and Huxley style modeling where the fraction of gates in a particular state has this functional form where there's a relaxation with a voltage-dependent time constant to a voltage-dependent equilibrium value. Essentially, you're assuming that the dynamics between the closed and open states of the gate, or the closed and permissive states of the gate, are mediated by rate constants that depend on voltage. You can write down the probability dynamics and using conservation of probability get a single ordinary differential equation, which ends up being linear. And so a little algebra can put it into this form that hopefully at this point is relatively familiar. Thanks for listening.